Okay, today we're going to talk about repeated sequences in our genome, mostly focusing on the amazing L1, its transposition, and its effects on our genome, which are many. So transposons in general are mobile genetic elements, and that they're mobile in the sense that they don't occupy any standard chromosomal location and that they can move around in the genome. And the process by which they do that is known as transposition. Now, there are plenty of DNA transposons, mainly in other organisms. We have a few of those, but the major part of the transposition in our genome has to do with retrotransposons, which are those that transpose by means of an RNA intermediate. They total more than 40% of our genome, amazingly. Okay, so here's a very dramatic figure, at least I think it's dramatic, which describes the sequence features of our genome, and it's kind of mind-boggling, really. That the protein coding exons, according to this, is a very tiny sliver of our genome. They're saying less than 1%. I think the standard belief is around 1.5%. But in any case, it's really very small. Other includes things like introns and intragenic sequences and whatever, centromeres, telomeres, whatever. But what's amazing here is the enormous percentage of our genome that's devoted to these various transposable elements, namely lines, signs, retroviruses, and DNA transposons. Collectively, they make up a humongous portion of our genome. And we'll talk about mostly the lines, which is L1 in our genome, because that's the one that most is actually known about, I think. Okay, so again, this is a summary, kind of an older summary, but it's pretty much in agreement with the previous, that we have the fraction of our genome of lines is 21%, 13% signs, retrovirus elements 8%, and DNA transposons only 3%. Okay, so the line is long interspersed nuclear element. Okay, so that's about, and those are non-LTR retrotransposons, which I will define in a minute. Okay, the full-length one is about 6 kb, but for reasons that we'll see, having to do with the mechanism of their transposition, most of the copies don't include the entire 6 kb. Right, the short interspersed nuclear elements in our genome, primary one is ALU, but also another one called SVA. Now these are very short, between 250 to 500 base pairs long, more or less. And these can move around in the genome, but they require proteins from L1 to do that. Okay, we have retrovirus-like elements, which are called the human endogenous retroviruses. These are the LTR type, meaning long terminal repeat, and that's characteristic of retroviruses in general. Now, we have a very few DNA transposons, and there are no known active ones. So the main players here are the lines and the signs, really. So in terms of some definition, classification, we have autonomous versus non-autonomous. Okay, so the DNA transposons transpose by a DNA type intermediate. They don't have any RNA intermediate required for them to move. The retrotransposons, however, are divided into a couple of different classes. The human endogenous retroviruses are considered as retroviruses, and those are the LTR retrotransposons, because their genomes are flanked by the long terminal repeat regions, which is the same on both ends of the element. So that's characteristic of retroviruses in general, as we'll see. Those are autonomous because they can catalyze their own movement. The reverse transcriptase is encoded in their genome, and that is involved in movement of these elements, whenever they do move. So that's the LTR part and the autonomous part. Similarly, line 1 is autonomous in that it has a reverse transcriptase, which is encoded by part of its ORF2 protein, but it does not have the long terminal repeats that are characteristic of the endogenous retrovirus type. So these are the lines, and they're autonomous and non-LTR. By contrast, the smaller guys, namely ALU, don't have enough coding capacity to encode their own reverse transcriptase, so they're non-autonomous. They're relying on the proteins produced from other elements in order to move around to the genome, and they are non-LTR also. So the autonomous ones encode their own reverse transcriptases. Okay, so these are the non-LTR ones as well as the LTR ones. And the non-autonomous ones have to rely on other elements, namely L1, for this purpose. Okay, so in all cases, the transposition process begins with the production of RNA by normal transcription carried out by the host cell RNA polymerase, not the reverse transcriptase encoded by the element itself. So we use Pol2 for the L1 and the endogenous retroviruses, and the signs use Pol3. But in any case, they are transcribed by our own RNA polymerase. Okay, so in terms of the reverse transcriptases, we know what those are. Those are RNA-dependent DNA polymerases. So what that means is they use an RNA as a template to synthesize DNA. And of course, they need to have primers for this purpose. So the ones that are most familiar, famous, are the reverse transcriptase encoded by retrovirus, but our own genome encodes actually, if you look at it right, 3 types of reverse transcriptase that are different. Okay, so our first reverse transcriptase is actually telomerase, if you think about it. This is an RNA-dependent DNA polymerase, and of course it's specialized for elongating the telomere repeat sequences. Now it's an RNP that it's got its own very specific RNA template and uses DNA at the ends of chromosomes as primer. So it counts as a reverse transcriptase because it's using an RNA template to create DNA. But our retrotransposons are a more important source of reverse transcriptase in our genome. There's the one that's encoded by L1, and there's the one encoded by the HERVs. So these are, of course, crucial for these retrotransposons to copy themselves to new locations in the genome. Okay, so here's another picture of the genome organization of these things, and we'll talk about them more later. The main one I want to talk about today is the L1, which is a non-LTR retrotransposon that encodes its own reverse transcriptase. We'll do the endogenous retroviruses next time. So here's another diagram of the genome organization of these things. The L1 being the one we're going to talk about now, where it has a 5' untranslated region, a protein called ORF1, a protein called ORF2, which is actually a protein that has more than one activity. It's one protein that includes an endonuclease, a reverse transcriptase, and also a cysteine-rich region. And it's followed by a 3' untranslated region and a bunch of A's. Okay, so just to emphasize how prevalent these are in our genome, I'm going to show a picture of the beta hemoglobin genes. Now, the beta hemoglobins are encoded by a gene cluster where the different forms of the beta globin gene that are prevalent at different times in our life, fetal and embryonic and adult, are arrayed in an array, a cluster, together. So the little lollipop things represent transposons. Okay, so within this picture, the human beta globin cluster, we have the epsilon, the 2 fetal types, the gamma, the beta gammas, etc. Then we have the delta, which is a minor adult form, and then we have the beta. Okay, now the introns are not shown. The little orange things are the whole gene, and the regions between the genes are just intragenic regions. We're not showing introns. But the little lollipop things emphasize how prevalent these transposons really are in our genome. The little green things are the ALU elements that are present, and the red ones are L1 or bits of it. So these things are just everywhere. Okay, so we're going to talk about L1, as this is the most prevalent and best understood. The life cycle, reverse transcription, but also some of the effects of L1 on our genome, which are pretty extensive. Okay, so first of all, with L1, most copies are incomplete, and they may contain mutations, or they very often. The important part is that they are 5' truncated. Okay, so that means that you have the 3' region of the element present, but it's chopped off before making its way all the way to the 5' end. So the elements that many of that you see are actually missing the 5' part, which includes, of course, the promoter. And this is an example of, well, that's a consequence of the way they transpose. There are, however, a small number, very small, of full-length active ones. These are called the source elements because they are the source of new transposition events. You have to have a full-length active L1 to move to a different location. And there's somewhere between 50 and 100 of these in each person's genome, but this varies. There are some very active ones that are called hot L1s. So even though a huge percentage of our genome is made up of L1-like sequences, the actual source of new transpositions or possible sources of new transpositions are relatively few. And it's very fortunate that they don't decide to transpose too often for us. Okay, in terms of the organization, the 5' UTR contains the promoter. Now this is pretty weird because usually transcription begins upstream of the transcribe region. Well, no, the promoter exists upstream of the transcribe region. But in this case, actually transcription begins within or at the very border of the 5' UTR. So this is a very unusual placement of the promoter. Okay, again, transcription is done by cellular RNA polymerase, and it produces a full-length RNA, which includes the 5' UTR, extends through the protein coding regions, ORF1 and ORF2, and the 3' UTR. So following transcription, it goes to the cytosol for translation to produce the ORF1 and ORF2 proteins, which are needed to complete the transposition process. Okay, so how it transposes, and this is somewhat weird. Okay, the full-length element is the source, and it needs to be transcribed from beginning to end to form a full-length 6 kb RNA, which then goes to the cytosol for translation of the ORF proteins. So we can produce ORF1 and ORF2 in the cytosol, where they will then associate with the L1 RNA itself, which seems odd. But it does. And then the collective forming RNP, the RNP then makes its way back to the nucleus, where the transposition process can occur. So what it needs to do when it gets back to the nucleus is to identify a target site where it's going to actually undergo what's called target prime reverse transcription to produce a DNA copy that will then become resident at the new location in the genome. And that is diagrammed. Oh yeah, I forgot to say this. Okay, the full-length transcript is bicistronic, which is very unusual because the two proteins are produced from the same RNA, which is, as you know, in eukaryotes, extremely strange. So the two proteins are named ORF1 and ORF2, and they were produced from the same RNA, which is a very unusual situation. But they do form a complex with their RNA and then go back into the nucleus where reverse transcription will occur after a new target site has been located. So the ORF1 is a small RNA binding protein. ORF2 is a multifunctional protein which has an endonuclease, which is needed to create a cut site where the new reverse transcription will happen. Then we have the reverse transcriptase, which will actually take the RNA and make a DNA copy of it. And then there's a cysteine-rich region. So they both are there forming the mRNP, and then they will undergo target prime reverse transcription, where the RNA is copied to form the DNA. Okay, this slide represents what happens during what's called target prime reverse transcription. So the target site on the DNA is sort of degenerate, but usually it's something like TTAAAA. And the endonuclease activity of ORF2 is responsible for cleaving the target site at both sites really in an asymmetric kind of way. So what happens is there's a target site cleavage by this endonuclease, and we envision this as peeling off of the target strand to align with the run of A's that occurs at the end of the L1 element. There's a bunch of A's there. So what that does essentially is provide a primer for the synthesis of the first strand cDNA. When you align your A's and then with your T's, they're base paired, that provides a 3' hydroxyl where reverse transcription can begin to copy the first strand of DNA, which is shown. Okay, so after that, there's going to be cleavage of the other strand to produce the structure underneath the peeled out thing. And so that leads to eventually target site duplication as well as synthesis of the other strand. So when the full-length L1, or whatever's going to get copied part of the L1, is finished, then the other end can prime synthesis of the second strand, because you made a cleavage at that location to allow that bit of single-stranded DNA to be available. So that's going to serve as our primer, and then that will extend across the whole element. So you can see where the target site, having been cut asymmetrically, is going to actually be duplicated in the process of this whole reverse transcription. So that will produce, if the reverse transcription process is complete, a new L1 copy at a new chromosomal location. And notice that since it's been done by an RNA intermediate, the old element is still there. So you will have accumulated extra L1s as a result of this process. Here's another picture, more or less, of the same thing. It doesn't really emphasize the target site, but that's something important to pay attention to. So the target site, and the fact that it makes two cuts, which are asymmetric, means that you're going to have this partially single-stranded bit that's ultimately going to end up being replicated along with the L1, and that's what generates the target site duplication. Now, importantly, reverse transcription doesn't usually make it all the way across the L1. It's 5' truncated, that is. So as a result, the transposed element has no promoter, can't produce its own RNA, and is basically not active in transposition anymore. It's dead on arrival. It's there. It's a new copy in the genome, but it's not able to catalyze its own transposition anymore. So the L1 RNA is actually destroyed during or shortly after the reverse transcriptase step. It's not clear where this RNase H activity comes from. And our host cell DNA ligase certainly seals the nicks, and it's not quite clear whether the second strand is made by our own repair DNA polymerase or by reverse transcriptase. People argue both ways. So here's another picture of target site duplication, where I think it makes it a little bit clearer what's happening. You have the asymmetric cut, and then peel the two strands apart. You can see there's sort of single-stranded, unpaired regions that are created as a result of that staggered nick. And that does get filled in during the process of replication of both strands, and that results in the target site duplication. So some effects of genome of L1 include clearly insertional mutagenesis, where L1 jumps into a gene and disrupts its function. This is sort of understandable. The other ones that are a little bit stranger are one that's called 3' transduction, which is also known as exon shuffling, mobilization of ALU elements, so that is the phenomenon of the non-autonomous transposition, where the ALUs are hijacking proteins from the L1 to catalyze their own movement. And then even weirder yet is the formation of processed pseudogenes. So here's a picture of types of mutagenesis, and this is very general. You can have a transposable element that produces an active element, okay, and it can jump into an exon, for example. So when in part number 1 below, we see the exon has now got a transposable element in the middle of it, and that will certainly disrupt the gene. You can also have a situation where you transpose immediately upstream of a gene, in which case you could bring the gene itself under the control of the transposable element itself. And other times you transpose somewhere and nothing bad happens. Okay, so in terms of genomic effects, there are verified cases where there has been transposition into genes that are driver mutations or causative of particular forms of cancer. So if you jump into the important coding regions of a tumor suppressor especially, then you're going to inactivate the tumor suppressor, and that's going to lead to cancer. You can also have events where you're activating a proto-oncogene, and again, that can lead to a cancerous phenotype. So there are very many documented cases where L1 has done something very bad by driving cancer formation by basically landing in the wrong place. Okay, so the next slide shows a very famous example of hemophilia, which also illustrates 5' truncation. Okay, so hemophilia is a blood disorder, bleeding disorder, that's caused by mutations in the factor VIII gene. Now, factor VIII encodes a blood coagulation factor, and in its absence, blood coagulation is extremely slow, leading to the bleeding disorder. So this is a case where a little boy has hemophilia, and his mother is known not to be a heterozygote. Now, in most cases of hemophilia, we have a heterozygous mother. It's on the X chromosome, and then male children get one of mom's X's, and either may or may not get hemophilia, depending on which X they get. But this isn't one of those. This is a different kind of case where it's a new mutation that arose from transposition of L1 in the mother, okay, or from the mother, let's say. So what has happened is that the patient has gotten a insertion of actually part of a 5' truncated L1 in his exon 14 of his factor VIII gene. And what that essentially does is inactivate the gene, which is why he has hemophilia. He's unable to coagulate blood in a proper order. So this is a new mutation caused by L1 jumping into that particular exon of this very important gene. An examination of the boy's L1, you can see that it's 5' truncated and that it has ORF2, but it's missing its promoter and it's missing the 5' untranslated region and it's missing ORF1. So that's an example of what this looks like. Okay, so you don't think that this is generally how these mutations occur. There are many mutations known in this particular gene that cause hemophilia, and it's just unusual that this is a new one versus an inherited one. Okay, L1 elements within introns can lead to 3' transductions, and what those are is downstream sequences are moved along with the L1. Okay, so this relies on the fact that the L1 can exist within introns of coding genes because their poly-A site is weak, so that you can get read-through of the L1 poly-A site to produce a rather elongated form of L1 that actually includes whatever's immediately downstream of it. So those downstream sequences become part of the L1 along with it, which means that they can get reverse transcribed at a new genomic location. So these are called 3' transductions. They used to be called exon shuffling. And usually, this is an unusual one that they're illustrating. They usually include only part of L1, sometimes no L1 at all. But the mechanism for their formation is reverse transcription of this hybrid RNA by the L1 reverse transcriptase in a target prime mechanism. Okay, so looking at what this looks like, we would have an L1 sitting in the middle of an intron, and that's not going to bother anybody most of the time. And you can see that the L1 poly-A site is present but weak, allowing read-through of the regular gene's transcription. Okay, so what happens in the case, it can happen, has happened, is that you start your transcription at the beginning of L1, just like you usually do. But then when, instead of stopping or polyadenylating at the L1 poly-A site, the RNA transcript extends through the intron into the next exon. So this exon part is now part of the RNA transcript and will be carried with it to a new genome location. So when the target site is chosen, you get target prime reverse transcription in the usual way, except it's going to be starting on the first gene, the source gene's poly-A, and including the source gene's exon 3. So what has happened is this exon 3 has been copied, moved from its original gene to another gene, basically extending that gene. Now in this example, we are including the L1 as well. But remember that L1 reverse transcription doesn't always go all the way. In fact, mostly does not go all the way. So it's perfectly possible to have this bit of exon 3 present with no sign of L1 at all, depending on where the reverse transcription actually stopped. So that's exon shuffling, or otherwise known as 3' transduction. Okay, so the next slide is a different version. Okay, and this has to do with ALU mobilization and also how you can form process pseudogenes. Okay, so usually when you have L1 RNA in the cytosol, it gets translated to form the ORF1 and ORF2 proteins, and then those associated with it to form the RNP that's going to get reverse transcribed eventually when it gets in the nucleus. Okay, but this sometimes doesn't happen. Sometimes you have escape and leading to ALU mobilization, which is to say the ORF1 and ORF2 proteins can bind to RNAs from the signs, or they can bind to and eventually reverse transcribe cellular RNAs as well. So when it's ALU that's being mobilized, when it's ALU that's being copied, that is non-autonomous transposition. But when you're copying a cellular RNA, that will lead to the production of what's called a process pseudogene. This is weird stuff. Okay, so here's one picture. There's a better one later, which basically shows the whole life cycle. You have your L1, makes your L1 RNA, goes to the cytosol, it forms an RNP, goes back to the nucleus, identifies a target site, and gets reverse transcribed. Okay, but sometimes these things go wrong, and the ORF1 and ORF2 proteins escape and bind to, instead of the regular old L1, either a cellular RNA or a RNA transcript from one of the signs, namely ALU. This picture's a little bit better, I think. Same thing. You've got L1 being transcribed, okay, but you can have what they're calling this RNP formation in trans. Okay, so in those examples, the ORF1 and ORF2 will bind to the ALU RNAs or to a cellular RNA, a regular cellular RNA. But after that point, the ORF1 and ORF2 proteins will carry out their functions just as if it were really L1. They will escort it back to the nucleus. It will find a target site, and it will then reverse transcribe either the ALU or the cellular RNA, depending on what it is captured. So once you get your ORF1 and ORF2 associated with the RNA in question, it goes back to the nucleus, finds a target, gets reverse transcribed, and appears at a new chromosomal location, even though it's not actually a line. Okay, so envisioning what this looks like, in the case of the process pseudogenes, if we have a RNA that's in the cytosol, lurking around, it came from a gene by the normal processes of transcription and splicing. So what this gene looks like in its gene state is, of course, it's got a promoter, it's got introns, and it's got 3' sequences, no poly A tail. But in the process of forming the mature RNA, of course, we don't have a promoter in our mature RNA. We don't have introns, and we do have a poly A tail. So the processed RNA looks different from the genomic copy. It doesn't look like a bona fide gene because it's missing things that genes have, namely promoters and introns. And genes don't generally have poly A tails. So what happens here, if we are going to copy one of these mature RNAs by a reverse transcription type process, we are making a DNA copy of that RNA. So the DNA copy that we make is going to end up being a process pseudogene after it gets delivered to a new chromosomal location. So these are recognizable. They're related by sequence to their progenitor gene, the gene from which they were made, the gene from which the RNA was made, actually, and in the sense that they've got recognizably similar sequence, but they are lacking introns, lacking promoters, and will contain poly A tails. There's also target site duplication that occurs at the location of the process pseudogene. Now, most times they're sort of inactivated slowly over time by the accumulation of mutations. So many times process pseudogenes can be recognized by the fact that they have incomplete open reading frames, they have frame shifts, they have stop codons in frame, and other types of mutations. So most of them are recognizably similar to their original gene, but contain mutations which make them obviously not active as genes anymore. So that's characteristic of most process pseudogenes. Sometimes they stay alive, in which case they could basically acquire other functions and are being conserved. But mostly they're not. Mostly they decay in a random sort of way. Okay. I think I probably said all this. Pseudogenes contain poly A tails, lack introns, no promoters, so they're not real genes. You can tell looking at them they're not real genes. And the fact that they have mutations in their open reading frames also. They're also often 5' truncated, as one might expect, because of the L1, like lack of processivity maybe of the RNA of the reverse transcriptase. Okay. One thing that's odd about processed pseudogenes, somebody did a very careful genome-wide survey of them in terms of trying to decide whether all genes are more likely to become processed genes, pseudogenes, or not. And the answer is no. If you look at the y-axis here, the number of human proteins, the 23,000 number is the number of human proteins which have no processed pseudogenes that can be detected. So the vast majority of our RNAs never did form a processed pseudogene, or if they did, they went away real soon. On the other hand, some genes have quite a few of them. Some have 60 or more examples of processed pseudogenes that are clearly related to the original gene sequence, but are now, of course, not real genes anymore by means of this reverse transcription process. So there's a wide variation, huge variation, in whether a particular RNA or gene will end up being a processed pseudogene or not. Okay, some famous ones. They tend to be highly expressed genes, which makes a certain amount of sense. If they're highly expressed, there's more RNA and more likelihood, therefore, that they would be the one that would capture the ORF1 or ORF2. But, for example, cyclophilin, GAPDH, actin all have quite a large number. Ribosomal protein genes are also very famous for having lots of processed pseudogenes. Strange phenomenon. Okay, so with all this activity here, why doesn't our genome totally explode with transposons? Well, fortunately, L1 is not expressed very highly, right? There's lots of silencing mechanisms that keep it limited in what it can actually do. So usually you have methylation of cytosines, deacetylation of promoter histones, silencing methylation marks, etc. So we're at war with this L1 in our genome. It's trying to transpose, and we're trying to keep it silent. So I think it's winning, actually. Okay, some of these mechanisms do get weakened in some types of cancer, and this can actually contribute at least to the possibility of new mutations which occur during cancer development. So if you're activating your L1, it might not be L1 itself that's causative, but its ability to undergo insertional mutagenesis and other sorts of events could probably do contribute to cancer development in some cases.